different ideas, different opinions about the topic. So because it's only a few of us, we're going to open the floor to questions as we go. So we may not cover all the topics, but it doesn't matter. What matters is to have a very good conversation and some ideas coming up. So here are the topics that we selected together last week. So we want to discuss the complexity of making antibody-drug conjugates and oligo. There are different challenges. We want to discuss money and profit, where we see the most profits in the future. We also want to discuss something that we had a good talk about over lunch, which is it one or the other? Is it one versus the other? What makes the difference? Why would you go for one and not the other? And finally, what's the outlook? If we had a crystal ball, because we have no idea, what would be the best bet for the future? So I suggest that we do a quick intro, and starting with you, Amit, and a brief description of your career, and then I'll finish, and then we go into the topics.
Yeah, so good evening, everyone, and thank you for coming. So my name is Amit Nayyar. I did my PhD. Okay, my name is Amit Nayyar, and I did my PhD in medicinal chemistry. After that, I did postdoc at National Institute of Health, USA, Bethesda. So I was doing research on tuberculosis there, and after that, I worked for a few companies, Korea, and then I moved to Canada, where I worked for a couple of companies. One of them was Thera Technology, where I learned ADCs and PDCs, how they work. And lately, I have been working as a techno-commercial with Cohance Lifesciences, where I am working as a business development guy, and I support their oligo and ADC business, actually, in East part of USA.
Mark Vermeulen. I am with Lupin Manufacturing Solutions, a subsidiary of Lupin. My career is pretty boring. 30 years in the industry, all with the same organization until the last year. Switched from Pfizer Center 1 to Lupin. I'm heading up the West Coast BD efforts for Lupin. Thank you.
Hi, I'm not Sophia Meng. I'm substituting for Sophia. I'm Eric Hohenschuh, Senior Director of Business Development for Porton. I have a 30-year history in the pharmaceutical industry. My degree is chemical engineering. My background is BD, both on the licensing and acquisition side, as well as the CDMO side. My experience is bridged small molecules and large molecules, and as a member of Porton, we are aggressively moving in the area of the new modalities, including ADCs and oligonucleotides.
Hello, everyone. So I'm Douville. I'm CatSci's head of business development for North America. So my background is organic synthesis. So after a PhD in total synthesis of a natural compound, I transitioned to the industry, initially at the bench doing medicinal chemistry for a couple of years. And then got drawn to the other side of the business, to sales and business development with Cycle, where my focus was at the interface of commercial and technical with purification technologies based on silica gel, so small molecules, but also peptides and larger molecules. And I transitioned to CatSci 6 years ago to lead business development activities in North America. CatSci is a small molecule PR&D company focused on activities like root scouting and process development. Started as a clear focus on small molecules, but we saw opportunities in oligonucleotides and peptides as more complex or bigger small molecules is how we see them.
And my name is Dom Hebrault. My background is chemical engineering. I have 25 plus years experience in process technologies as they apply to big pharma. I define process technologies as anything that goes beyond the round bottle flask, which includes high throughput screening, lab automation, process analytical technologies, flow chemistry, spray drying, milling, crystallization, solid form science. And so I had process engineering team for those process technologies right now at our headquarters at Veranova in Devon, Massachusetts. We are a mid-sized CDMO focusing on complex chemistry, small molecules, but also tides, which include peptides and oligonucleotides. And we have announced an investment of $50 million in antibody-drug conjugates with a groundbreaking event in June this year. And so we're going to open with the first topic. So here are the topics: complexity, profit strategy, and outlook. I suggest that we go around the table, starting with Amit, give you some thoughts, couple of minutes. If you want to react on the spot, please do so. Please contradict us, give a different opinion. That's what the magic is. So complexity, ADC and oligo, if you want to give your thoughts on that one. Amit?
Yeah, so I mean, ADCs are evolving in last 20 years. We can see that there are almost 14 to 15 ADCs which are evolved, which have been approved actually. And if you really look at it, it's very complex. It's not a small molecule because you have to have a payload which is cytotoxic. So you need not only facility, you need capability also. So I believe that it's a very, very complex process where you need a linker, you need a payload, so you have to club them together. Then you have to do conjugation, and then you do drug substance to drug product. Now, every step you need specialized manpower actually, because for handling HP API, you need a facility where OEL level has to be 5 or so. Then when you do with linker payload, and then you go for conjugation, so you need MAb capabilities to produce MAb also. So it's like you have to have 3 to 4 to 5 different capacities under a single at a single place. And major problem with the ADCs right now is controlling DAR, because you want from one batch to another batch uniformity, and you want same data for everything. So I think manufacturing is a little challenging. And there are ways, people are coming up with the new ways to counter those issues, and new developments are going on. So that's beyond the scope of today's discussion, but yes, it's a very complex process actually. And CMC is so difficult that it requires a lot of, you have to have a very skilled manpower actually. And that's why big companies have their own capabilities, but small companies or small biotechs, they have to rely on CDMOs, which are really knowing how to do that actually. And for oligos, similar thing actually. In oligo space also, the major drawback there is the monomers are really tricky. Some of them are like 27, 28 step synthesis. So once you deal with such monomers, and when you bring them together to make your oligo, there can be truncated oligos in the mixture. So that brings another complexity. So that's how I will say it.
Mark?
Good. So for LMS, I think we're focused in on the ADCs. I'm not saying there's anything wrong with oligos. It's just a matter of our priority right now is on ADCs. The complexity is where we thrive. And I think, as you look at that and across the production facilities anyway, the complexity breeds the containment as well. And, it's something that we're used to and understand. And again, it aligns with the vision, that we're looking to go.
Sure. Many of the same comments, but I would add to the complexity of ADCs, really thinking about the heterogeneity of the large molecules, in addition to the complex structures, but the instability of these molecules due to hydrophobicity. There are also the same problems that the others have mentioned with the OEB5 warheads typically used, and even beyond OEB5. So the ability to work with these highly potent molecules at the same time that you're managing the challenges of working with the large molecules. On the oligonucleotides, really from our perspective, the challenge is not so much the assembly and production of small-scale batches of the oligonucleotides, but of the ability to scale up that process and the complexity and cost of the equipment to make commercial-scale oligonucleotides. The bridge from early stage deliveries for phase 1, phase 2 clinical trials to commercial trials is a much larger gap than in other parts of our industry.
So we have a bit of a different perspective at CatSci because we are strictly a CRO, so we don't do manufacturing at CatSci. And being traditionally, or coming from a traditionally small molecule background, we saw the evolution of the complexity in small molecules moving, beyond a rule of 5. And when we started looking at ADCs, it was clear to us that we would not be doing the full process for ADCs because that's just beyond our scope of expertise, and being a smaller organization, we need to be really, really good at what we do. And so when we started thinking about ADCs, the opportunity for an organization like ours was really around the payload and the linker, which we consider to be small molecules. They're just more complex small molecules, essentially. And that was essentially our thinking also with when we built our oligos capabilities and our peptide capabilities. I mean, they are covalent bonds that are made by chemists, and we are chemists. So this is something we figured we could make a meaningful contribution, not only on developing processes to synthesize those sequences in traditional, solid phase supported way, but also thinking about how do we solve or how do we bring about the next generation of synthetic strategies for these modalities? Because the reality is there's just not enough acetonitrile in the world right now for all the solid supported synthesis that is being considered. So we need to think about all of these things in terms of complexity. So from our point of view, we need to be innovation partners. We need to be keeping pace with the industry on the innovation side, and that's how we're approaching this question.
Yeah, thank you. So at Veranova, if we were students, I would say we would have a major in ADCs and a minor in tides, to say so. So why do we take that direction? Because we have a 25 years legacy of controlled substances. So we already have in place the pressure cascade, segregated room, and unidirectional flow in the building. We have a holistic approach to risk management, not a one-off. So we have already the capability, the experience. So making important drugs is kind of natural. We also have a legacy of complex chemistry. So making the warhead and the linker, assembling the two is pretty natural to us from an expertise and capability standpoint. We haven't had a biological activity. We are building that today. So you don't need a BLA, a biological license, to actually make biologics. But we can manipulate antibodies. And we are starting a team, small scale, doing bioconjugation. So we are going full speed into ADC that we see as an opportunity based on a strong experience. So on the peptide, on the tide side, it's a little bit different. So we now have a history of making oligonucleotide peptides. It requires a ton of expertise in very complex, many-step chemistry. You've got to consider that for 20-25 residue tides, you have capping, uncapping, oxidation, reduction step. You probably have 50 or 60 or 70 steps, and you want to make as much use of automation as possible. And so the reason we are doing that is that we have major brain power acquisition in the company. We had our CSO, CTO coming from back, and we have major high-level people in the company that came from peptide suppliers. And so basically we have the knowledge and the brain power from them to build capability in that direction. But again, like I said, it's a minor in our equipment, and we are building all the automation that we have on site right now to develop that at small scale GMP side. Maybe it's a maybe it's a good time to react on your side before we move to the next topic. Any question, any experience that you want to share? The complexity of ADC and oligonucleotides. Okay, so I suggest to move to the money aspect of it. Where does everybody see wins on the long run for each of the two categories, for CDMOs and innovators? Where is the money to be made in the future? And I guess the natural answer is to say in both directions. So go ahead, Amit.
So this is my personal opinion. If you look at biotechs, they have to have a very specific goal, actually, so they cannot broaden their horizon in different therapeutic areas. So usually companies either focus on ADC-based therapeutics or oligo-based therapeutics. But as a CDMO, if you really ask, I think both are holding a great potential, because ADCs are being utilized for countering the cancer. And as you see, like, it's a multi-billion-dollar industry, and there are at least 200 clinical trials going on. So in coming years, till 2030, we will see that many more ADCs will be approved, and there is money. And if it is developed by a small biotech, then they definitely need support from CDMOs. So there is a big potential for CDMOs. Similarly, in oligo space also, it depends on who is the innovator. If it's a big pharma company, they have their own assets. But if it's a small company, they have to rely upon, their aim is to make money by curing the disease, of course. So they are working in oligo spaces in ASO or siRNAs or mRNAs. But when it comes to scale up, they have to rely. So I think for CDMO, it's wide open, game is wide open, and both modalities are picking up big time. And we are trying to treat diseases by using oligos, where we don't have a real, treatment available with small molecules. So I think there is a big potential in both areas.
I think it's good. I think the one piece that you look at is what you're good at, right? And I think that that also plays into the fact. But from a looking to the future from a monetary perspective, of course, the rate of growth right now is in the ADCs, right? You see that growing rapidly, a little bit more rapid than the potential for the oligos. Not saying, again, not bashing either side. I think there's a misnomer for this versus piece. I think it's really just talking through it. But when I'm looking at it from the perspective of LMS, again, we are fully involved with the ADCs and focusing on the ADCs. Haven't ruled out oligos. It's just not one piece that today we have focus on. Today we're focusing on the things that we have, our capabilities and what we're good at.
Yeah, I would concur. ADCs are growing faster right now than oligos. I think the fairly recent data has a CAGR of about 30% on the ADCs and about 16% on the oligos. We see value in both areas and see opportunities for the oligos to continue to grow, especially as they gravitate from being single applications for rare diseases to multiple applications for chronic diseases over time. So there is an opportunity for growth in both. We also see an opportunity for different CDMOs to approach this question differently. We see that for the smaller CDMOs, there may be a better choice to focus on one or the other and work on technical barriers, become the best at certain specific technical aspects of the production. We see larger CDMOs as being fully justified in pursuing both areas and looking to mitigate market risk. That no one really has a crystal ball about which of these areas is going to ultimately be successful. Both of these are strong areas for growth for the CDMO industry because of the important medical benefits these products are bringing to patients, and the success in cancer from the ADCs, the success in rare diseases from the oligos is truly phenomenal. And so pursuing these areas for the CDMOs, I'd say the other thing that in addition to the growth of the revenue, the big pharma companies, the big customers, don't really have an advantage in capability or versus the CDMOs at this point in time. These are new areas. Nobody has a incumbent advantage the way that the big pharmas may have in small molecules.
Thank you. I feel like I'm representing the small guys here because I'm sitting at this table with very big companies. And like I said, at CatSci, we're somewhat smaller. We're 150, 160 people. And I think all of you guys at this table have, multi-thousand people in your companies, respective companies. So companies like ours, when we look at the profitability of those modalities, we kind of see two angles here. First of all, we have to position ourselves to be a partner to the industry, and we're going to make money if the industry makes money, right? And the only way to do that, especially working with biotech, is to unlock the full value of an asset. And to do that, there's two things to consider. One of them is is the scalability aspect. So is this asset fully scalable? Can it be made on scale reliably, high yield, high purity, reproducibly in a robust way? So that's one thing we look at when we develop processes at CatSci. And the other thing is building the CMC package, because again, the full value of the asset will be tied to the CMC package. If you don't have that, then the asset doesn't stand on its two legs, and there's a risk. There's a risk for the asset value, and there's a risk when it goes to manufacturing. So our role in a small company like ours is a bit more focused on enabling that CMC package and enabling that manufacturing process to be reliably done on scale. And the only way for a company like ours to survive and thrive is to deliver really great science. And I think that's going to be key for the profitability of CDMOs and CROs in this space. The only ones who are going to be able to win are going to be the ones who are able to be problem solvers, deliver good solutions, and accelerate time to clinic, essentially.
Yeah. So I see that after some challenging beginnings for ADCs, I think the plane has taken off already. So we all know there is money to be made in ADCs. For oligo, it's more recent. I would say that the plane is on a running track or at gate still. So we don't know which plane is going to go farther. So eventually it's difficult to anticipate how much money oligonucleotides are going to generate now, whereas we know that ADCs are going to be profitable and generate a ton of money. You can say that oligonucleotides probably address more therapeutic areas than ADCs. So intuitively, you may say actually in the future they may develop wider and then potentially becomes very profitable and a good direction to take. As far as the wins, it depends on who you are. If I were to create a biotech company in the Boston area tomorrow, I would probably go in oligonucleotides more than ADCs. Because I have limited funding, and it seems to me a promising area to innovate. If I'm a big pharma, maybe ADCs are more interesting. For CDMOs, it depends on the CDMO. Sure, you don't want to put all your eggs in the same basket, but that also depends on how many eggs you have, right? Mm-hmm. So I would say if you're a major player, and we have some on the table here with me, maybe investing in both is a good idea. If you're a public company, you can issue share, raise money, and maybe invest. If you're a tiny company, VC funded or emerging CDMO technology oriented like CatSci, maybe you want to pick and choose. And so you can make ADC. You don't have to make the whole assembly. You don't have to be worried about the drug-antibody ratio. You can also focus on the warhead and the linker and offer that. It's a smaller market, but that's can be very profitable, and there is a need in that direction. So you can pick and choose. For us, we are in the middle. We are mid-size, so we are pursuing both directions. I guess we have one that's for the short term, the ADC for the next 5-10 years. We know we're going to generate revenue, and we have strong assets because of what was in place already back 10 years ago on site. For oligonucleotides, we are at the very beginning on investing and looking at what we can offer to the market. Yeah. So in case you haven't heard, I'm going to try to repeat the question. What are the opportunities for ADCs outside the oncology market? Right? That's the question. Absolutely. And that's something we discussed. Go ahead.
Yeah. So actually, in ADC field, people are coming up with different types. So in apart from oncology, ADC is picking up because in ADCs, it's a targeted delivery. So these molecules, whatever you see today, whichever are being delivered using ADC, there were some issues with those molecules actually. So like if you see XTCAN or DXT, they were in XTCAN especially was in clinic, but it didn't make it to the market. But it was toxic. So using ADC, they could bring it selectively to the site of action. So that's the way they developed it. Now, apart from this, people are working on degraders, protein degraders, where the molecules are again not following Lipinski rule of 5. They are beyond that, and they are delivering those selectively using ADCs. They are using even immunomodulators also to enhance immunity, and I think in radionucleotides are also being delivered using that. Now, as you said, like in infectious diseases, the thing is where we want infection is throughout the body actually, but here we are targeting a targeted stuff. So if infection is in the lung, maybe we have to first develop antibody for that and then try to. So it's better like the PKPD such that it is distributed uniformly throughout the body, and then you're seeking. So that's how I feel.
What is the ratio of, maybe the panel can answer that, ratio of oncology and non-oncology?
I think for now it's 10-1, roughly. But you have antibody oligonucleotides in development, so that could really change the paradigm. I let my colleagues share their thoughts on non-oncology ADC opportunities.
I mean, sorry.
Go ahead.
Sorry, please.
Yeah, I mean they're out there, and it thinking of rheumatology as one area, it's fairly natural in the ADC to picture the ADCs following the use of the antibodies, which are the key targeting agent. There are obviously going to be areas that people in this room have not imagined yet where these molecules will be used, but I think it's also when you go to the oligonucleotides, the initial rare disease focus of these makes sense for a number of sort of economic and commercial reasons. And as the technology expands and grows and becomes more capable and more affordable at lower cost therapies, further applications will be explored and used.
So I think you touched upon something that's getting where the therapeutic dose is getting smaller, right? So there's less and less needed of this product. So from a CDMO perspective, we deal with volume. We want lots of volume. ADCs are going to be
But just to clarify your question, and then I'll let my colleagues answer. You make it sound like there is a psychological high price per kilo that max that you can charge. Is that what you mean?
To a certain extent, yeah.
Okay.
You don't need that much, right?
Okay. So you cannot charge $100,000 a kilo.
You can, but you only need 10 kilos.
Right. You had a
My name's Mark coming out. I've got a sort of related question. So you talked about CAGR growth, which obviously is a great top line. What is the capital investment that a CDMO or CRO needs to make to be a serious player? And the second question, sort of naturally, when do you reasonably expect to make a return on investment? Probably comes back to the question you were asking.
Yeah.
Next. When do you reasonably need that to sort of start to pay off? Large pharma, they're not just going to carry can for all this right now. Presumably the biotech market's not in high growth mode, so that's not going to pay right now. So this is going to come down to your shareholders who are PE funds putting capital behind those propositions. I'm interested to know what sort of investment needs to be made for each, and how do you deliver that? When do you need to deliver that?
So we can bundle that up actually and address all the questions at the same time. So the question is, how much can you charge per kilo to basically make the investment profitable over a reasonable time, which would be what, 3 years, 5 years at the most? I'm not an investor, so I'm not sure, but let's say 3 years. I don't have the answer. Does anybody have that one?
No.
I don't have an exact answer, but I can answer that at Porton that the strategy is to move into these areas in a stepwise, logical fashion where the investments that we're making are following the needs of our customers and are consistent with the economics that they see in the business. The question about, how much can you charge per kilo? I mean, if you asked, biotech people this, back in the 80s, there were people were outraged that TPA was going to be $1,300 a dose, and look where we are now with the price of some of these monoclonal antibody therapies. So I think for Porton, our strategy in both of these areas is to invest one step at a time, consistent with the needs of our customers who are able to put projects on the table. I think most CDMOs operate in a very similar way.
Yeah.
Can I extend my question by one point? What's the lead? So from the point in time that you have a client who says, what we like to do X, whatever that might be. Obviously, to put the capital investment to get to a position to deliver that need. Are you able to move? She's able to move fast enough, but do you need to be making bets?
Oh yeah, totally.
And
No, I can address the question. So you're not waiting for a client to knock on the door without any capability to start building. That would not work. The client would not come to you. So it's a bet, yeah. You do the investment, then you go after clients. We say, hey, look at that, what we built for you guys, even though that's not true. As far as ROI and return on investment, I think that depends very much on whether you need to raise funds for that or whether you're self-funded. So in our case, we are self-funded, so that's our money. We do what we want with that as long as the board agrees with that. So it depends a little bit on the company. And I'll stop there now.
And it's not just CapEx, it's the people also. And we're talking about very, very niche skills. So finding the right people to operate, those reactors is probably trickier than buying the equipment. I think you can source the equipment pretty readily, or within a certain lead time. But finding the capable workers to operate that is another tricky thing to figure out.
And is the expertise for oligonucleotides?
Yes. Yeah. Because you can build a scalable facility for oligonucleotides pretty quickly, because the volumes you need ultimately are pretty low. And there are, for example, the Oligosynth series of reactors from ACTA are built to go from lab scale to pilot scale pretty quickly in a very linear way. But anybody with a few $1 million will be able to build a facility and buy those equipment, but it doesn't matter if you don't have the right people in front of those systems running them.
And also, I cannot speak right now of some of the oligonucleotide modalities. Like what we do is mostly focus on RNA interference, which is a 20-25 residue peptide, but people also make longer peptides for RNA silencing, and there can be above 50 residues. And then you have all the diagnostic market also. We don't sell right now to the diagnostic market. We may in the future, but we have to build that knowledge, that connection, because there are different people in different companies. So if anyone has any experience, want to share some thoughts on the alternative oligonucleotide market, the one that we don't talk too much about, like diagnostic and silencing, that would be interesting. So we have touched already on the two topics at the bottom. The strategy, should we do one or both? It depends. As always, it depends. And what's the outlook without a crystal ball? Which one will predominate? It's difficult to say, and that depends to whom also. But yeah, absolutely. Please react. We have another 5-10 minutes. We pledge to be available until 11 p.m. if you guys want. And then they shut the hotel. And
Done. Good.
have we covered all the questions you guys had? I'm trying to recap actually the three that you said. Return on investment, it was that price per kilo. I don't know about the price per kilo. It's a little bit unfair though to ask for a psychological price per kilo if you compare the 10-step, 12 steps. That was the case 10 or 20 years ago on traditional small molecule. And maybe we have complex chemistry that go into the 40 and 50 steps. And so, and the price does not increase linearly with the with the number of steps, right? It's more complex than that. So, yeah, I don't know. It doesn't feel right. Have you got that kind of feedback from clients?
No, just in general.
Okay.
Saying from a CDMO perspective, volume, fills your reactors.
Okay.
Turns the lights on. But in specialized medicine like this, you're using much less material.
Right.
Which is significantly more expensive, like you're saying. Maybe $100,000 a kilo, maybe
Right.
$200,000.
Yeah.
But you need significantly less volume.
Yeah.
So you make $2 million on that.
Sure. The operation. So.
And revenue, you're not making a lot of money.
True. On the upside, so on our side, we have labs, of course, where I work. We have kilo labs. We go up to 100-liter scale reactors. And in the pilot plant, we have mostly 200-gallon and a few 750 that are used for commercial scale production. And so it used to be that the 200 gallons would be used for CDMO work, like clinical development phase 3. Today, we barely ever touch them for clinical development. We use a kilo lab. So the kilo lab was used for validation before scale up. Today it's used for production. But the operation cost of kilo lab is a lot less than plant scale. So sure, you spend more money on the number of steps and making the API, but you spend less on non-proportional cost by using the kilo lab instead of the pilot plant. So again, I'm not the CFO of the company, but I would imagine that you spend more on one side, spend less on the other side, on labor cost, for instance.
Yeah, one more question. So, is this just going to be a big commoditized space, or is there going to be room for somebody to get an intellectual position that will allow them to dominate, even if it's one particular vertical in the manufacturing space?
Oh, on the oligo, 100%. So the oligonucleotide synthesis, the way we do it, is not good. So we waste a ton of solvent, it's unsustainable, and it's very costly, we're very wasteful. And so the room to improve oligonucleotide production is gigantic. We were discussing over lunch today, we've been doing solid phase traditionally, we're looking into liquid phase. And if you do solid phase, how can you actually accommodate the expansion of the bed so that as it expands and contracts, as you grow the chain, you can accommodate for that and make it better. And there was a AICHE talk from Lily actually talking about countercurrent washing at the end to reduce the amount of solvent to remove the junk actually from the product. So there are rooms for improvement. So there is a ton of engineering in oligonucleotide production that we can capitalize on. I don't know if it's IP or inside knowledge, that I don't know, but yeah, absolutely. Oh yeah. Any oligo geeks?
Throw some light on regulatory acceptance of ADC because you need to have huge clinical trial to validate the efficacy and also how difficult it is for any organizations to approve.
I think for that, it is like for ADCs, regulatory approval is very similar. Only CMC can be a little complicated. That's how I feel. Because in CMC, you have to make sure that payload which you are taking is according to ICH guidelines. Linker also. So it's not a single factor. There are many factors which you have to. So there is a very complexity in CMC. But overall, when it comes to clinic, it's based on the efficacy, whether you see the overall outcome or not, whatever trial you have decided for. But CMC is very tricky, actually. So you have to provide a lot of documentation for each and everything. And for payloads like whether it is a topoisomerase or something else, you have to follow the guidelines which are for small molecules. And same is for the linker. And then once conjugation is done, then it's it will be treated as a biologics. But I mean, tricky part is CMC. That's how I see. But
So you prefer ADC because of less side effect. Okay, so
yes.
it has to be improved and how to improve.
So it's a patient outcome. I mean that.
So you need to have huge clinical trial. It is not normal.
Can you
I'm not
maybe repeat
sure.
the question so that everybody understands what question is?
So the question is like he's trying to ask like how will you evaluate the outcome of the ADC program actually? How will you evaluate?
I think in general, in pharmaceutical development, the more substantial the clinical efficacy of the drug, the smaller the clinical trials can be to absolutely demonstrate effectiveness of the drug. That was what was so exciting about ADCs when they first came forward. You had oncology trials where it was rare to ever see a response in a patient in these trials, and seeing all of the patients respond. I think the complexity in the development of the ADCs is really more on the CMC side than on the clinical side.
So in terms of how you when we're talking about that, you mentioned there are 200 drugs in clinical trial ADCs, and maybe I'm not sure on the Oligos. So this becomes a more niche kind of expertise, or this is something which is evolving and this is
So the question is whether, so we have 200 drugs in clinical development for ADCs. How many do we have for oligonucleotides, and do we expect that to grow? So the count is a little bit different in the sense that companies like Sarepta or Dyne that develop a technology platform around a family of oligonucleotides, and then they diversify based on the technology. So once they have the technology in hands, they can actually replicate that across many candidates. So it's a little bit a different way of generating innovation in oligonucleotide and ADCs. On ADCs, actually, you have innovation on the antibody and the linker and the warhead. Even though on the warhead, actually, there is a little bit less innovation because they're usually already existing molecules. Rarely do we develop a warhead for an ADC. They are usually readaptation of existing ones. So it's difficult to answer your question. Is the number for candidates for oligonucleotides going up in the future? If it does not, I don't really know why it would not. So the answer is yes. Is that going to be above 200? I don't know, but based on the way it works to generate innovation, I believe it would be in the thousands, actually. Yeah. More than the hundreds. And then I stop there.
Just to add to that, so in clinical trial, the most effective, important aspect is patient selection. Like when you are doing that study, patient selection is most important because many times the patients which are participating in clinical trial have already seen different kind of therapies. And if, let's say, we are dealing with the ADC with topoisomerase inhibitor, and the patient has already seen irinotecan as a drug during course of chemotherapy, then there are chances that already those, there are more transporters in the body and cancer has seen. So it's very tricky, but I mean, patient selection, and on top of that, how you see the partial response and clean response. So there are many factors actually associated, but many times it's not that you have to, there are 100 patients, then everyone, you have to see, they should be clear of infection, the cancer should be cleared from the body. But even if you see partial response or clean response, how the tumor is behaving, based on that, recommendations are made. So it's beyond the scope of this one, but that's how you see those outcomes actually. That's all I'm saying.
Hey, I would love to continue the conversation all night, but we have to respect the schedule of the hotel and leave the room because they have to clean up and everything. So we can continue the conversation, but not here. Maybe around a piece of pizza or something outside. Yeah. Our names are on the agenda. You can connect on LinkedIn. Maybe you want to spell out your name before we finish so that people can contact you. Because you're not on the agenda, are you?
Yeah. Eric Hohenschuh, Hohenschchuh.
But feel free to connect on LinkedIn. That would be fantastic.